Hello, my name is Vaughn Cooper. I'm a professor at the University of Pittsburgh in the School of Medicine, and I'm really grateful for the invitation to share this presentation with you at ISME 2026. And I'm really sad I can't be there in person, and I want to give a special thanks to Susie Wiles for the invitation to be part of this symposium on microbial science education. Again, I'm really looking forward to interacting with you, although I'm sad I can't be there in person with you this time. This work is about our program known as Evolving STEM, which aims to build science identity for students in grades 6 through 12 through authentic microbial evolution experiments happening in the classroom, not a summer camp, but actually in their ordinary schools. And this work's been led by Dr. Abby Matella and more recently by Maya Mahler in our laboratory, as well as a huge team of students and postdocs and volunteers over many years, and couldn't be possible without them. So our motivations really start from the observation that there's been a revolution in technology to enable education for STEM, but that's largely ignored the life sciences despite huge need. There's broad recognition that hands-on learning with technology can really engage students at a really young age and make them feel like they're part of the process of learning in science, technology, and education. But that's really lacking in most of the life sciences. And then the second piece, my favorite topic, evolutionary biology, is unfortunately not well appreciated, certainly in the United States, in part because the instruction is boring. And much as I love learning about Darwin's finches and the adaptive radiation that Darwin discovered and many other examples of ongoing evolution, there's nothing like an experiment to show evolution. And microbes in potentially offer that solution. So my aha moments that got me here really began when I was a PhD student starting out in Rich Lenski's lab, working with his long-term evolution experiment that's been ongoing since 1988. He's been studying, his lab's been studying, and now successors have been studying the evolution of these 12 populations of E. coli. Half of them have a white marker, half of them have a red marker that allows you to mix them and study how an ancestor is outcompeted by the evolved population. And that kind of blew me away, that I could see changes in frequency associated with fitness in real time. And so that started the hypothesis that maybe learning evolution might be more effective when we can see and measure it in real time. Essentially, what we're looking at, is changes in frequency over time. So that's a real example of fitness and ongoing evolution. But if we're just looking at changes in frequencies of markers, that's a relatively subtle concept that may not be accessible and may require more time than most students and teachers have. The next kind of revolution for us was when I, started studying biofilm forming bacteria, namely those that cause opportunistic infections in persons with cystic fibrosis. The best known of these is probably Pseudomonas aeruginosa, or perhaps Staphylococcus aureus. I was studying one called Burkholderia sinusopatia, which is an opportunistic pathogen associated with particularly severe infections of persons with CF. And to study how they form biofilm and cause these biofilm-associated infections, we created a really simple model of putting a plastic bead in a test tube and then only transferring the bacteria that attached to that bead to the next round, selecting in the sense for reversible stickiness over days of passage. And what we saw after only really a few days of passage is the bacteria rapidly produce more biofilm. So you can see here, there's increased biofilm at the air-liquid interface of these tubes that are grown on a slant or roller drum. And there's also, if you zoom in, chunks of biofilm on those plastic beads in those test tubes. More surprising to us is when we plated those tubes, we saw repeatable patterns of phenotypic diversification that in turn was heritable. So each population gave rise to 3 different morphologies: studded, ruffled, and wrinkly. They had different biofilm-associated phenotypes. Collectively, when you mix them, the biofilm was synergistic. That synergism was associated with greater antibiotic resistance, greater resistance to invaders. Was ultimately, when we applied genome sequencing, the underlying genetics was associated with adaptations that we know are associated with a range of biofilm and growth traits and resistance traits. And many of those are actually found repeatedly during chronic infections where you apply genome sequencing to study adaptation to the cystic fibrosis airway. So that sort of once pointed out that we could develop models of biomedically relevant processes simply by selecting on biofilm and using appropriate nutrition to model aspects of the infection environment. And that would connect this process of rapid adaptive diversification to biomedical problems. And so just circling back, our hypothesis is that learning evolution is most effective when seen and measured in real time. And our approach now was that if we could do these processes studying biofilms, you could see strong, rapid adaptation, but also a process of diversifying selection where students, any students, myself included, could see the process of adaptation at the phenotypic level happening quite quickly. And so our original goal setting out was to adapt this Burkholderia process to using a safer organism, potentially for the classroom, and to demonstrate that evolution and diversification could happen quickly, safely, and reliably within a week, and then ultimately connect what the students are seeing with the relevance of the processes we're studying in our laboratory, to teach them essential biotechnology techniques, to increase their learning of key next generation science standards in evolution and heredity, also microbiology, to teach them about microbes themselves, to promote student inquiry, and ultimately to scale all of these processes. And so what we do is introduce that, the fact that most bacteria actually live in biofilms in a well sort of structured matrix, and biofilms themselves generate heterogeneity that may lead to some of this diversification. Biofilms are associated with a range of ecosystems like plants, found in streams. They're associated with a range of properties with hosts, both beneficial and pathogenic, including in humans. They can form sort of slime and pipes for deleterious consequences. They are kind of the model of what dental plaque is, in fact, the model of biofilm. It's a polymicrobial structured community. And then also biofilm-associated infections are well known, especially in persons with diabetes, like foot ulcers. So those provide the context. How does it work? We start out with a clone of Pseudomonas fluorescens, strain SB-W25, pretty famous around the world, and also really well studied there in New Zealand at one point. Still is, I think. And then that clone is propagated by students from over the course of 1 week. And by the end, from Monday to Friday, the tubes change, and the students can observe more biofilm at the air-liquid interface. And then they plate that tube on after the first day. They see no variation. They plate that tube at the end of the experiment on Friday. They come back in the following Monday, and if they're fortunate, they'll see plates with diverse colony morphologies. And they can actually pick those colonies and streak them out to show that they breed true. As I said before, they're not just producing, say, one new colony morphology, they're producing diverse types, indicating there are multiple niches or solutions that allow the bacteria to adapt. So again, the question really was, excuse me, that the students learn better from this experiment. We were able to partner with 2 amazing teachers back in New Hampshire, where we started out teaching 9th grade honors biology, and they split their classes into an experimental group and a control group. And then they reversed them, meaning the experimental group got our experiment as the curriculum, and then the control group got the ordinary curriculum, and then they swapped. And we measured their learning on the same next generation science standard targeted assessment over time. And what we found is that our curriculum improved learning by about 50% more than the regular curriculum. But group 2, which had the regular curriculum, following getting our curriculum caught up. And so group 1 and group 2 were not statistically different from one another at the end of the experiment. And so, in short, they learned better, and this is great. But then we started to ask, in the following couple of years, how does this experiment influence their overall sentiment towards STEM? And what we found is a number of gains, some of them statistically significant, like I enjoy learning science, I enjoy learning technology, I'm good at learning technology, I will make it into a good college and major in an area needed for a career in STEM. And some of those were really more strongly biased towards the girls than the boys. And we've seen this trend repeatedly, that we see general gains, but especially stronger gains among the girls more than the boys. So at this point, my lab moved from the University of New Hampshire to the University of Pittsburgh. And that caused us to, sort of reshape our teams, recruit new people to help, and start to grow in a new environment. And so our new goals were to expand to more schools, districts, and states. Really need to do that. We needed to simplify and streamline our programs into kits. We had to build rigor and flexibility in curriculum materials. Really wanted to aim to create a community of practice among educators so that educators could teach other educators and help them along the way. Obviously needed to build a sustainable funding model, and that continues to be an adventure. And then we also wanted to get the students connected to our research, to have student research in their classrooms help us generate scientific discoveries, ultimately making many more scientists. So first we were able to distill our program down to a kit that we can put in the box, and then we could also get support to get a bunch of durable goods like incubators, shakers, vortexes, and pipettes and so on that we could loan out to regional schools. That ultimately, over the last several years, has led to pretty significant expansion, as you can see, across the United States. We're in about 12 states currently, and in a range of mostly middle and high schools, but we've also been adopted in a number of colleges and universities and more autonomous adaptations. We're now engaging over 2,000 students annually. You can hopefully see from these slides where we're at nearly 80 cumulative teacher partners and around 55 school partners over time, with around with fewer currently active, although that changes from year to year for a variety of reasons. And so that growth has allowed us to dig in more with scale about how Evolving STEM influences science identity. So in a first pass of this data, we really focused on Pittsburgh Public Schools, a highly diverse and largely underserved area for authentic research of all kinds. And what we found is this majority underserved population showed significant gains in questions like I am a scientist. So you'll note that they start out quite low on this scale, but by the end of just the experiment, only 2 weeks, we repeatedly see significant gains in that response. Further, I often ask questions about science. Now, it's not all roses. In this particular survey, there was a somewhat decrease. I want to find out more about science. Perhaps the experiment sort of showed them that research is challenging, or there's some setbacks associated with the process of doing research. Turning more to the labs, we also found significant gains in confidence about their ability to do labs, like I know more about than my friends about doing science labs. So I'll talk more about some assessments coming up, but we've also been able to really expand the science. So that means that by getting the students to pick mutants, they can give those mutants to us. We're quite good as a lab at genome sequencing. We can sequence those genomes quite quickly, turn them back around, provide the raw fast cues to the students and their teachers, and then in collaboration with middle author bioinformatics, we're able to build a web app of the best-in-class software BreeSeq developed by my friend Jeff Barrick, formerly a postdoc in the Lenski lab, who's now the steward of the Lenski long-term lines, really helping to direct that experiment going forward. And this amazing web app allows for really the latest method of detecting variants of mutants in these genomes, and the students can plug that data in and quite quickly get an answer. What is it, what's the mutation in my mutant that makes my colony look different? And here you can see at the top, the cause of one of those wrinkly looking colonies is a mutation in the wrinkly spreader phenotype, WISP gene F, a well-characterized circuit by the Rainey Lab and many others shows excuse me, that leads to a cascade that ultimately upregulates the signal cyclic diGMP, in turn ultimately producing more cellulose and a wrinkly colony phenotype. So students can rapidly discover phenotypes. They can also pick them. We can genotype them. And over the years, we can accumulate a number of data points that allows us to now assemble a research study on the genetic causes of diversification in Pseudomonas fluorescens, which we concluded was being maintained by this hypothesis that there are in fact multiple niches in these biofilm models that causes the maintenance of diversity by negative frequency-dependent selection. This work was recently published in Applied and Environmental Microbiology, led by Dr. Metella, involving many of us in the lab. And I'm just going to give you a couple of quick snapshots. Here's just showing a pileup of mutations in this WISP pathway that I was talking about, but also a bunch of mutations in this YFIRNB pathway, also in this well-characterized Moray gene. These three gene circuits are illustrated here sort of as cartoons, all operate by signals largely in the periplasm that are transducing cellular stress to activate cyclic diGMP synthesis. And that cyclic diGMP synthesis gets upregulated and in turn activates the synthesis of biofilm-associated polymers. What's important about all of these is they've all been characterized in the related pathogen Pseudomonas aeruginosa as drivers of biofilm-associated adaptations during infections. We've also characterized some less well-described causes of adaptation, including mutations in this FUZY gene that's been characterized before that alters LPS, but also in a newer regulator of cyclic diGMP that we named BMO for biofilm and motility regulator. It too has a transmembrane domain that activates cyclic diGMP. And finally, a number of interesting mutations that alter disulfide bond production. And that alteration of disulfide bonds alters how proteins interact in the periplasm, in turn also likely activating cyclic diGMP. So in the last bit, I want to tell you a little bit some new work that we did about it created a new curriculum that's really allowed us to expand to younger student groups, but also enable different forms of inquiry. And this curriculum was quite literally inspired by the book and now movie Project Hail Mary. And here's just some snapshots from the movie. Here's Dr. Grace from the book and movie, played by Ryan Gosling in the movie, looking at this predator that he discovered that's eating the extraterrestrial parasite that's killing many suns. And when we were listening to this, my son and I, we just wondered about whether we could maybe adapt this predation experiment or this notion of studying a predator so that my son Harlan, shown here, could do an experiment that wasn't just simply a riff on the same old EvolvingSTEM biofilm selection experiment. So what we did was grab a bunch of mutants that students had isolated that differed in their biofilm production. We spread them on plates, and then we put the predator that we knew that was available to us, provided by my friend Terrence Levin's lab, Dictyostelium discoideum, the predatory amoeba, in the middle. And hopefully you can see here is one of his plates where there's a clearing in the agar caused by the predator. So certain types were eating the bacteria more than others. The fact that this experiment worked well enough for his seventh grade science project led us to bring it back to the lab, to riff on it, to see if we could make it more reproducible. And fast forward, now thousands of students have used our predation curricula. And now they've really motivated our research. And so just to give you a sense, we've learned that mutants producing different biofilm amounts and types defend against dictyostelium predation. So I'm playing a video right now, and you can see that two of them are lac marks, so they're blue. And now, hopefully you can see in the middle, different mutants are being eaten at different rates. But over here at the right, that WISP-F, wrinkly mutant, is barely being eaten at all. And the BMO mutant over here has been eaten almost as much as the ancestor. The MUC-A or mucoid one is sort of intermediate. So what we're finding is that this wide variation in mutant types in their ability to defend against predation suggests that bacteria are pre-wired to defend themselves against predation. And maybe that's not so surprising because they've been encountering phagocytic predators for probably about 1 billion years, and yet we actually don't understand that very much. And so we're using dictyostelium discoideum, or dicty, to model phagocytosis. We found that a single WISP mutation can increase predation resistance by up to 20-fold. And that suggests that Pseudomonas in general are pre-wired to escape from phagocytosis, literally only maybe one SNP away from a really big gain that protects them. However, these mutations are either costly, and so there's a pretty strong balance between defense and growth that we're recognizing. And so that we've got a nice new research program, really outgrown from science education, that is driving a lot of our work in the lab. Thanks to former PhD student Nanami Kubota, who built a nice R-Shiny app. Students can now upload their data, which is literally just the area of bacteria consumed in their experiments, and so they can compare among mutant types and among different schools. That way students can see that they're part of a community of learners, that different classrooms have different experiments and so on. And so we can come back now and we can ask, well, how do the biofilm and predation labs compare for their ability to change student identities? And you can see that, ask questions on science, more of a gain for the biofilm experiment than for the dicty experiment. I am a scientist goes up for both. That's great. We're making students more likely to feel that they are actually scientists. The biofilm experiment may do a little better job at this notion of, I can become good at science. I know more than my friends in terms of their labs, stronger for the dicty experiment than for the bead experiment. So these provide some nuance. And of course, this is, again, we're growing this data set over the years. We want to know how many of these trends are reproducible over time and what are the factors that influence these motivations. But these kinds of tools are allowing us to refine both the science experiment, but also the way we deliver the education to ultimately create more gains across all these categories. So in short, I hope I've shown you that our work in studying how bacteria adapt to new environments, and particularly adapt to established biofilms associated with disease, has translated to the production of curricula. And those curricula ultimately help students see themselves as researchers. And the connection of their research to our research makes them feel like they're part of the overall biomedical process. Collectively, we can hopefully learn how to better treat infections and save lives, and student experiments are contributing directly to our overall research goals, and we in turn are helping them join us in the next generation. Thank you again for the invitation, for the opportunity to join you. This work's been supported by the National Institutes of Health with a Science Education Partnership Award. Up until quite recently, it's been supported, or it's at the end of its support, to train teachers through this amazing NSF BioRETS program. And here are a couple of photos of the lab with many missing here. And thanks again.